Hello students, welcome to EPG Pathshala. I am Dr. Shaukat Meet from Hamdard University, Delhi. Today we are going to discuss about a module named Alkaloidal assays under paper Pharmaceutical and Biological Analysis. After completing this module, we should be able to understand following. Number 1, definition, properties and extraction of alkaloids. Number 2, general methods for the estimation of alkaloids and in plants and in preparations. Finally, we will also discuss some of those individual assays of alkaloids. Let's first understand what are these alkaloids. Alkaloids are naturally occurring nitrogenous organic substances which contain nitrogen as the part of their heterocyclic ring structure. These are produced by plants as secondary metabolites. They are weakly to strongly basic in nature. Biosynthetically, they are derived from amino acids. They have diverse physiological actions even at very low doses. So these parts of its definition make these alkaloids. So in addition to these carbon, hydrogen and nitrogen, they may also contain oxygen, sulfur, besides some other elements like chlorine, bromine and phosphorus. Alkaloids are produced by a variety of organisms, not only by the plants only. So some bacteria, fungi and even animals produce some of those alkaloids. They can be purified from the crude extract or from these organisms by acid-base extraction. The alkaloids have a wide range of effects, wide range of pharmacological actions, such as quinine. These are cinchona alkaloids. They are antimalarial, as we understand. So we also have like ephedrine as anti-asthmatic agents, quinidine, which is another alkaloid present in cinchona, which is anti-arrhythmic in effect, while as who doesn't know about the analgesic activity of morphine, which are present in opium. Let's now understand the principle of alkaloidal assays. So the proportions of total alkaloids in plant materials and the preparations that are made from these are usually determined by process which are actually involves the interplay of the properties of alkaloids. So it initially involves the separation of alkaloidal content from the non-basic contents. So for the purpose of following general properties of alkaloids are made use of. Number 1, the alkaloids we understand are usually sparingly soluble in water but readily soluble in non-polar organic solvents like ether, chloroform, benzene and light petroleum. The other property, second property of alkaloids that is used is that alkaloids combine with acids to form salts. These salts are usually freely soluble in water but only sparingly soluble in organic solvents like ether and chloroform. The third property of alkaloids is that alkaloids are liberated from their salts by alkalis. The methods employed involve the use of pairs of immiscible solvents. So we may use during this process immiscible solvents such as water and ether or water or chloroform. Now let us discuss the general procedure of alkaloidal assays. As I told you earlier that they are based on the general properties of alkaloids. First, a weighed quantity of material in suitable subdivision, means in powder form or in small divisions, or a weighed quantity or a measured quantity of a preparation is treated with a suitable alkali to liberate the alkaloids which are generally present in combination with organic acids like meconic acid, acetic acid, etc. Number 2, the free alkaloids are extracted by means of a suitable solvent, for example an ether or chloroform. The solution is shaken with acidified water in a separating funnel. The alkaloids then pass into solution in aqueous layer as salts, leaving non-alkaloidal extractive matter in the ethereal layer. The next step involves the aqueous layer, where the aqueous layer is separated. It is made alkaline and it is shaken with ether or chloroform again. The liberated alkaloids pass into this solution in the organic solvent, which is then separated and evaporated. So the organic layer is separated and it is evaporated to dryness. A little bit of alcohol is added towards the end so as to get rid of total volatile matter. The alkaloidal residue finally is weighed or it is determined by titration. This table presents a list of drugs containing alkaloids along with their botanical origin, the part used and the total alkaloid content needed. Let's see, like first belladonna leaf, which is Atropa belladonna. The leaves and the flowering tops in the dried form are used as official drug, and it should not contain less than 0.35%. The other entries are like stramonium. Datura stramonium is the official drug. The dried leaves and flowering tops with branches along with branches are used and should not contain less than 0.25% of total alkaloids. Another drug of the same class of solanaceous alkaloids, for example Duboisia, very important drug. It is obtained from Duboisia myoporoides. Dried leaves are used and contains a very high content of alkaloids from 2 to 4%. Apart from these solanaceous drugs, we have other drugs like Synchronous, which is obtained as the dried bark from Synchronous succirubra and contains 6 to 10% of total alkaloids. Opium is another drug, which is actually the dried latex obtained from the incisions on the unripe capsules of Papaver somniferum, and it contains not less than 10% of morphine. Rauwolfia, as we know it as Indian snake root, it is Rauwolfia serpentina. The roots and rhizomes are used that contain somewhere around 0.8 to 1.3% of total alkaloids. In the same way, agate, which is actually a parasitic fungus growing on the rye plant. So dried sclerotia contain several of those agate alkaloids. List of important alkaloids along with their molecular formula and molecular weight. So you can see at the top of it there is atropine. It is a propane alkaloid. Its molecular formula is C17H23O3N. The other propane alkaloids like apotropine and hyoscyamin are also in the list. Two strychnine glycosides found in Nux vomica like brucine and strychnine down in the list, they are present in Nux vomica. Codeine, morphine are the two opium alkaloids which differ in one extra methyl group. So we have morphine C17H19NO3, while as codeine is C18H21NO3. Other on the list are quinine, reserpine and nortropine. Let's now discuss the estimation of alkaloids in detail. As I have already mentioned, most of the methods involved in the determination of alkaloids are variations of the general processes of extraction from natural material by solvent extraction in presence of alkali, followed by acid extraction, and finally solvent extraction from alkaline solution. So let us go into the details of these processes. Number one, extraction. As we understand that ether or a combination of ether and alcohol or ether chloroform mixtures are used for the extraction of alkaloids from the material. The process of extraction can be percolation or continuous extraction. Benzene is another choice which can be used as a solvent as it leads more cleaner extracts. Preliminary treatment of drug with menstruum containing alcohol should be restricted as it ultimately leads to extraction of higher portions of chlorophyll, resinous matter and water-soluble amines and saponins. The extraction can also be carried out using ammonium hydroxide. The next process is purification, which is generally carried out by alternative extraction between chloroform and dilute sulfuric acid. The alkaloids are generally quite stable to hydrolysis in dilute acid solution and in neutral or slightly basic conditions. The time required for their extraction comes well within the limit of its stability. The free bases are relatively stable in solution of organic liquids. The time during which alkaloids are permitted to remain in contact with alkali must be reduced to a minimum. So, the alkaloids are taken in aqueous solution by salt formation, the salts of like sulfate salts. Next comes the evaporation. In this step, the final organic extract is evaporated to dryness to remove the ammonia and heated for a period till all the volatile amines are removed. It has been shown that evaporation of alkaloidal residues from alcohol and heated at 105 degrees Celsius for about 5 hours produces no deterioration due to loss of basicity or lowering of recovery on titration. Once this extraction is complete, we will also like to ascertain whether the extraction has taken place completely. So in that case, we may need some of those detection methods. So these are some of those general tests for alkaloids to ascertain the completion of extraction. As we understand, Dragon-Droff's reagent consists of potassium bismuth iodide. It gives a reddish brown precipitate if the sample contains alkaloid. In the same way, Mayer's reagent, which is potassium mercury iodide, produces a cream colored precipitate, while as Wagner's reagent, which is iodine potassium iodide solution, it produces a brown precipitate. Hager's, which is a concentrated solution, saturated solution of picric acid, yellow crystalline mass. There are some specific methods. As for propane alkaloids, we have Italian-Morin reaction, while as for meconic acid, which is closely associated with morphine, there is special test, which is the treatment of sample with 10% ferric chloride solution gives a blood red color. This is a test which indirectly gives the confirmation of the completion of extraction. The determination of alkaloids can be done by volumetric titration or by weighing the final residue. For volumetric titration, an accurately weighed crude drug is macerated with ammonia and water. The crude drug is then poured, it is filtered, and it is shaken with chloroform. The chloroform portion is then extracted with acidified water in 2 portions. The acidified water solution, acidified water is, the portions are mixed and the alkaloid is released, liberated by treatment with ammonia, and finally the residue is taken in an organic solvent which is evaporated to dryness. To this residue, this residue can be weighed for the residue calculation, or it can be subjected for volumetric titrations. The volumetric titration can be of both types, aqueous as well as non-aqueous. In case of aqueous titration, the purified alkaloidal extract or residue is dissolved in a measured volume of 0.02 normal H2SO4 solution in presence of 3 to 4 drops of methyl red as indicator. The excess of sulfuric acid is measured with 0.2 normal sodium hydroxide by back titration. The total content can be determined using the formula given, like in this case it is for atropine. The volumetric titrations can also be carried out in non-aqueous medium. Here the alkaloidal residue of the crude drug is dissolved in 5 ml of chloroform and 5 ml of acetic acid is added. Then the solution is titrated with Nb50 perchloric acid in presence of gentiana violet as indicator. Then the calculation of alkaloidal content can be done using equivalent factors. Here the formula is for the determination of atropine. Let's now go to the study of individual alkaloids. First one is Synchronous alkaloids. There are more than 40 alkaloids that are found that have been reported from Synchronous. The Synchronous, as such, as we understand, these are the dried bark from the stem or the root of Synchronous succirubra, Synchronous calicea, Synchronous officinalis, or Synchronous ledigriana or its hybrids. The average content of Synchronous alkaloids varies from 6 to 10%. Quinine, quinidine, synchonine, and synchonidine are the major alkaloids that are present. So the formula on the right hand side gives the two portions of the Synchronous alkaloids. The portion in the red is a quinoline nucleus, while as the portion in the black is a quinacridine nucleus. So the slide also shows the diagram of Synchronous bark. So just to mention that totacoin was a standardized preparation containing at least 15% of quinine and 55% of other crystalline alkaloids. Earlier it was used extensively before the isolation of these quinine alkaloids in pure form. Now let us discuss the assay of Synchronous alkaloids. The total alkaloids in Synchronous can be calculated or estimated by the following method. So it involves few steps. The first step is we mix the powdered bark with a mixture of 7.5 ml of strong lead subacetate and 12.5 ml of water, and we allow it to stand for 1 hour. Add 50 ml of ammonical ethanol. We mix it well and allow it to stand for another hour. Transfer to an apparatus for continuous extraction in a Soxhlet apparatus or a percolator with a little more ammonical ethanol, and we let the extraction take place for about 4 hours. Evaporate the greater portion of ethanol. Add 10 ml of 1 normal sulfuric acid and 40 ml of water. We heat it and allow it to cool. Filter through a tightly packed cotton in a separator and extract it with portions of mixture of 5 ml of 1 normal sulfuric acid and 15 ml of water. The mixed acid solution is pooled and it is made alkaline with dilute ammonium solution and shaken with successive portion of chloroform. Wash the chloroform with little water, evaporate to dryness, add ethanol 5 ml towards the end and re-evaporate. Dry the contents to a constant weight at 105 degrees Celsius. Now let us discuss the assay of Synchronous alkaloids in Synchronous extract. A Synchronous extract is a soft extract made with strong alcohol containing at least 10% of total alkaloid. So procedure goes like this. Mix about 2 grams of extract, this soft extract, with 5 ml of water and 5 ml of ethanol, 95%, and add 1 ml of 1 normal HCl to the mixture. Number 2, we extract the mixture with chloroform in successive proportions by shaking 20 ml each time twice for 2 minutes, washing each extract with the same 5 ml of 1N H2SO4. Retain the two aqueous solutions separately and add to each solution 2.5 ml of sodium hydroxide solution. The extract, the mixture is then extracted with successive proportions of chloroform by shaking for 2 minutes until the extraction is complete. Combine the extracts, wash with little water, reject the washings, evaporate off the chloroform. Add 5 ml of 95% ethanol, re-evaporate and dry the residue of total alkaloids to a constant weight at 105 105 degrees Celsius. Emulsification may take place with chloroform while shaking with chloroform. This can be avoided by adding little bit of glycerol in proportions of 0.5 ml to a maximum of 2 ml, and if it doesn't help, a little bit of ethanol can also be added. In the same way, let's understand the assay of synchronous alkaloids in the form of quinine hydrochloride solution. From quinine hydrochloride solution, the alkaloid is liberated by addition of NaOH and extracted with successive quantities of chloroform, each chloroform solution being washed successively with the same portion of water. In this way, loss of quinine in water washings is reduced to negligible. The mixed chloroform solutions are evaporated, a little dehydrated alcohol being added towards the end. So the residue is dried and weighed. Quinine dihydrochloride injection and quinine bisulfate are assayed in the same manner. Quinine sulfate is sprangly soluble in water and is dissolved with the aid of dilute sulfuric acid, and the alkaloid is then liberated by addition of NaOH and extracted in the same way as quinine. Quinine sulfate tablets and quinine bisulfate tablets are extracted with water and dilute HCl, and from the filtered solution, the alkaloid is liberated by ammonia and extracted with chloroform. The liberation with ammonia and extraction with ammonia are also used for quinine and urea USP and quinine and urea injection USP. The opium alkaloids. Of the many alkaloids that are present in opium, the most important ones are morphine, nascopine, papaverine, thebaine, narcine, and codeine. Good quality opium should contain not less than 10% of morphine. As we understand that opium is the air-dried milky exudate from the incisions on the unripe capsules of poppy, Papaver somniferum of family Papaveraceae. Opium contains more than 40 alkaloids, which are usually combined with specific acids like meconic acid or other acids like sulfuric and acetic acid. Of these, nascopine, papaverine are comparatively simpler derivatives of isoquinoline, as is shown in the lower panel. On the other hand, morphine, thebaine, and codeine are more complex, which have these phenanthrene nucleus shown in the middle of the right hand side. Morphine has another first associated with it, that it was the first alkaloid to be discovered isolated in the crystalline form. It is the chief alkaloidal constituent which is responsible for its medicinal activity of opium. The assay of opium alkaloids involve first, like morphine sulfate, from a solution made alkaloid by addition of sodium hydroxide, and non-phenolic alkaloids are removed by extraction with chloroform. Alcohol and chloroform are then added, and the sodium derivative of morphine is decomposed by means of ammonium sulfate. The free morphine is extracted by successive quantities of alcohol mixture. The mixed extracts are filtered and evaporated. The residue is dissolved in a measured volume of 0.1 normal HCl. The excess acid is determined by titration with 0.1 normal sodium hydroxide. The equivalent factor for the calculation of morphine sulfate will be 100 ml of 0.1 normal acid will be equivalent to 1/20th morphine sulfate. Next is morphine hydrochloride. For morphine hydrochloride solution, the alkaloid is liberated in an aqueous alcoholic solution by means of sodium bicarbonate and is extracted with a mixture of chloroform and alcohol. The washed chloroform extract is evaporated. The residue is boiled with measured volume of 0.1 normal HCl, and after cooling, the excess acid is determined by titration with 0.1 normal NaOH. Codeine phosphate tablets. The tablets are first extracted with 0.5 normal sulfuric acid, and the alkaloid is liberated from the extract by ammonia and it is extracted with chloroform, washed. The extracts are washed with and evaporated. Complete removal of volatile matter is ensured by adding alcohol and evaporating to dryness. The residue is then dissolved in alcohol, treated with measured volume of 0.1 normal HCl and diluted with water. The excess acid is back titrated with 0.1 normal NaOH. The equivalent factor is given next to it. For codeine phosphate tablets USP and codeine phosphate injection USP, the same method is followed using N/50 sulfuric acid. Compound codeine tablets and soluble compound codeine tablets are assayed for acetyl salicylic acid, phenacetin, and codeine phosphate. The alkaloid is liberated by means of NaOH, extracted with chloroform, and removed from chloroform solution by 0.1 normal sulfuric acid solution. Thereafter, the procedure is same as that of codeine phosphate tablet mentioned above, using 0.02 normal sulfuric acid. Next set of alkaloids are that from Rauwolfia. At least 20 alkaloids have been isolated. The most important of these are reserpine, desreserpidine, and methoserpidine. Reserpine is obtained mainly from Rauwolfia serpentina or Rauwolfia vomitoria, belonging to family Apocynaceae. Nowadays, the reserpine is obtained by synthesis mainly, synthetic source. It is a complex base containing naphthodiazofluorine ring system, as shown on the right hand side. It acts as a tranquilizer and is used in relief of hypertension. The official recognition for Rauwolfia has been made in the Indian Pharmaceutical Codex 1953 and British Pharmaceutical Codex 1954. The latter specifies that the drug contains not less than 0.8% of total alkaloids, while as per IPC, the range is from 0.8 to 1.3%. Though only 3 alkaloids, reserpine, resinamine, and desperidine, have been shown to possess the desirable hypotensive properties and have also been shown to yield. There are other near about 20 alkaloids in the molecular weight range of 280 to 635. So there is a large diversity of alkaloids found in Rauwolfia. For the Rauwolfia alkaloids assay, the reserpine undergoes many color reactions, and that makes the basis for their identification test and assays that are used. For example, the reserpine gives a greenish yellow color with sodium nitrite in dilute mineral acid, and this is used in the assay where the measurement of extinction at a particular wavelength is carried out. The reaction with nitrous acid is not simply formation of nitrosamine with secondary amino group, since it has been shown that 11-methoxy group and diazofluorine moiety are essential for production of color complex. Among other alkaloids from Rauwolfia, resinamine, which also contains 11-methoxy, also gives this reaction, while as desreserpidine, which is 11-D-methoxy, reserpine, it does not give this reaction. Reserpine injection and reserpine tablets are assayed by the same method after extraction with chloroform. Now let's discuss about these agate alkaloids. Agate consists of dried sclerotia of parasitic fungus Claviceps purpurea of family Claviceptaceae that arises in the ovary of rye plant, which is scale cereal of family Graminae. So on the right hand above side, we see an ear of rye plant and with these black sclerotia. So this is a fungal infection that has replaced the ears of rye plant. The preparation of agate have long been used in obstetrical practice for inducing uterine contraction and controlling hemorrhage. There are about 10 agate peptide alkaloids. For example, to name a few: ergotamine, ergocine, ergocrystine, others. There are 5 pairs of isomeric alkaloids. Agate alkaloids are substituted amides of lysergic acid. So alkaloids of agate exist in stereometric pairs, as I told you earlier. The levorotatory alkaloids are pharmacologically active and they have configuration of lysergic acid, while as dextrorotatory alkaloids, which are inactive, have the configuration of isolysergic acid. Regarding the assay of agate alkaloids, ergotamine, methyl ergometrine, and ergometrine are determined in their salts and preparation by the colorimetric method. Ergometrine is a relatively simple amide, as you can see. It is derived from lysergic acid and 2-amino-1-propanol substitution. Its assay involves a reaction with 4-methyl-aminobenzaldehyde, which in presence of sulfuric acid and traces of ferric chloride gives a blue-violet color. The color is matched against that given by a standard of ergometrine maleate. Ergonavine maleate USP, however, is assayed by addition of ammonia to a solution, extraction of ergometrine with ether, evaporation of the extract, the solution of the residue in 0.02 normal HCl, and back titration with 0.02 normal alkali. Bromophenol is used as an indicator here. Now let's discuss propane alkaloids. We understand that these propane alkaloids, they are mainly present among the members of Solanaceae family. The tropanes are most well-known alkaloids found among its members. The plants that contain these substances have been used from centuries as poisons. However, many of these substances have invaluable pharmaceutical properties. Some of the examples of propane alkaloids are scopolamine, atropine, hyoscyamin, etc. Propane alkaloids are the basis of over 20 active pharmaceutical substances, which have varied uses such as medriatics, antiemetics, antispasmodics, anti-anesthetics, and bronchodilators. There is a list of these plants along with their names, which actually contain these propane alkaloids. So there is a detailed module for these propane alkaloids separate under the same paper. The chemical structures of major propane alkaloids: hyoscyamin, atropine, and scopolamine. These propane alkaloids, they are known to prevent binding of acetylcholine to its receptor, and as a result have effects on heart rate, respiration, and functions in the central nervous system. Among these, scopolamine acts as an antagonist at both peripheral as well as central muscarinic receptors. Thereby it shows varied effects. The assay of propane alkaloids involves a simple process of dissolving a weighed quantity in neutralized alcohol, followed by adding a measured volume of 0.1 normal HCl. The excess of acid is determined by titration with 0.1 normal NaOH. An equivalent factor of 1000 ml of 0.1 normal acid equaling to 1/10th of this atropine is used. In case of atropine sulfate, the substance is dissolved in water, the solution is made alkaline with ammonia, the liberated alkaloids are extracted with chloroform. After removal of chloroform, the assay proceeds on the similar lines as above. While as for atropine sulfate tablets, a weighed quantity of powder tablet is subjected to prolonged treatment with cold water containing sulfuric acid so as to get whole of the alkaloidal salt in solution. The solution is filtered into a separating funnel and made alkaline with ammonia. The liberated atropine is immediately extracted with chloroform. After evaporation of the washed chloroform extract, the residue is dissolved in N/50 HCl and the excess of acid is treated with N/50 NaOH. For hyoscine hydrobromide determination in hyoscine tablets and hyoscine injection and hyoscine eye ointments, a similar method as mentioned above for corresponding atropine preparations is used. So this is an USP method for the assay of atropine, where atropine is accurately weighed and dissolved in 50 ml of glacial acetic acid and 1 drop of crystal violet is added. It is then titrated with N/10 perchloric acid solution to give a green color endpoint. A blank determination is also performed and any necessary correction is made accordingly. Another USP method is based on TLC, where the test solutions are made in a concentration of 20 mg per ml and 1 mg per ml. The reference standard of atropine sulfate USP containing 24 mg per ml is also prepared. Then 25 microliters of first solution containing 20 mg per ml of atropine solution, 1 microliter of second containing 1 mg per ml of atropine solution, and 5 microliters of reference standard. These are applied to a suitable thin layer chromatographic plate, which is coated with 0.5 mm layer of silica. The mobile phase that is used for development is chloroform acetone diethylamine in the ratio of 5:4:1. The potassium iodoplatinate is used as a spraying reagent. Then the RF values are compared. The RF value of the principal spot obtained from each of the test solutions should correspond to that obtained by reference standard. No secondary spot obtained from the first atropine solution should exhibit intensity equal or greater than the principal spot obtained from the principal spot. So students, let us now summarize what we have learned from this module. We now understand that alkaloids are naturally occurring basic nitrogenous organic substances which essentially have nitrogen as the part of their heterocyclic ring, or in some cases it may be present outside the ring. So they are physiologically active in very small concentrations. The other thing we know is that the alkaloids are classified on the basis of their heterocyclic ring present. So they may contain a quinoline nucleus, an isoquinoline nucleus, or phenanthrene or naphthodiazofluorine nucleus. Most of the methods for the estimation of total alkaloids include the extraction from natural material by a solvent extraction in presence of alkali, followed by acid extraction, and finally solvent extraction from alkaline solution. Both aqueous and non-aqueous volumetric methods are extensively used in the alkaloidal assays. Various colorimetric, gravimetric, and instrumental methods are also used for the estimation of alkaloids in plant materials and their preparations. Many sophisticated techniques are now employed, many of those are hyphenated techniques, for the accurate and picogram scale estimation of these alkaloids in plant material preparation and even in biological fluids. Thank you.